Hello everyone, my name is Qingxi. I'm a master student from Stanford University. Today, I'm honored to share the work NanoSpring, reference-free lossless compression of nanopore sequencing rates using an approximate assembly approach. This work is done together with Shuban Changdak, Yifan Zhu, and Professor Saki Weissman. As a background, the nanopore sequencing, specifically the Oxford nanopore technology, has seen increasing adoption. As shown in the picture below, there are several advantages of nanopore sequencing. It is portable, real-time, it supports long reads, and native DNA and direct RNA sequencing. However, the error rate of nanopore sequencing is also high. A typical human whole genome sequencing experiment can produce hundreds of gigabytes of data in FASTQ files. Therefore, the compression of those files becomes very important for storage. The general purpose compressor like gzip cannot exploit the redundancy in the reads. There existing specialized read compressor for short reads, but they do not work for long nanopore reads. Therefore, for our work, we developed the NanoSpring, which is a lossless reference-free compressor for nanopore sequencing reads. As shown in the schematic here, NanoSpring could compress a nanopore sequencing rate of size 84 gigabytes in whole human genome file into a compressed archive of size 7 gigabytes. This compressed archive is 3 times smaller than that using gzip. The computational requirements of NanoSpring are practical, although it uses more time and memory than previous tools. The NanoSpring is freely available online at the link in the box. Next, I'm going to talk about the methods of NanoSpring. The figure here shows the overall process. NanoSpring first indexes the reads using Meng hash, which enables efficient lookup of reads overlapping a given sequence. Meng hash is a technique in computer science for quickly estimating how similar two strings are. Then, NanoSpring picks an arbitrary read to start with, and then greedily keep finding matching reads. The matching reads are found using Meng hash index, and the alignments are done by using widely used aligner Minimap2. Next, I will give a more detailed example of the consensus graph. In the figure shown here, the nodes in the graph represents the basis, and the edge in the graph stores the information about the reads passing through that edge. The weight of an edge is the number of reads passing through the edge. Initially, the graph is just a line graph consisting of a single read. As reads get added to the graph, these reads lead to branching out from the line graph due to presence of arrows. The consensus sequence represents the path with the highest weight. In this example, 2 reads are shown here. The read with the path in the green has a substitution compared to the consensus sequence. The read with a path in the blue is reverse complemented with respect to the consensus sequence and has a deletion and an insertion. The consensus sequence is shown in red. With the consensus graph, we can encode the 2 reads, and the table here shows the encoded streams of the 2 reads. Note that the arrow position is 0 indexed and delta coded, and the arrow bases need to be stored only for insertions and substitutions. For example, for the first read, the starting position is 1 relative to the consensus sequence. It has an arrow position at 2. The arrow type is a substitution, and the substitute base is T. The RC flag indicates that the first read is not reverse complemented. In the end, all the encoded streams are compressed with an effective general purpose compressor BSC. Next, I'm going to share the experimental results for NanoSpring. In the table here, we compare NanoSpring with popular general purpose compressor gzip and a state-of-the-art compressor eNano. We test on 4 different whole human genome datasets from different samples and with different coverages. Overall, NanoSpring achieves close to 3 times improvement in compression over the state-of-the-art compressor. Note that the compression results for the first dataset is significantly worse than the other 3 datasets. Although NanoSpring still outperforms the gzip and eNano, The reason is that this dataset was obtained using an older base caller with much higher error rates. Given the improvement in the base caller quality over the years, we can expect the performance of NanoSpring to improve further in the future. For now, NanoSpring could only compress the reads in FASTQ file. Therefore, for the future work, we plan to incorporate NanoSpring into a complete compressor, which could also handle quality values and read identifiers in a FASTQ file. Thanks for watching till the end. Feel free to leave a message if you have any questions or comments. Also check out the paper at the bioRxiv link here if you are interested in more details.